Thus far, we have exclusively discussed the genetic engineering of prokaryotes. Engineering eukaryotes is also possible, and there are specific biochemical and cellular processes present in these organisms that enable additional types of genetic devices. The most obvious of these differences is the potential for multicellularity. One of the most useful qualities to multicellularity is that it enables the genetic encoding of macroscale morphology through developmental processes. Daisy Ginsburg is an artist who has speculated on the future of synthetic biology and the potential for encoding the biological production of complex consumer products. Here she imagines how individual components of a gardening device could be produced in engineered organisms and then assembled into an herbicide sprayer. Though such applications are beyond the current capabilities of our field, eukaryotes and developmental processes are nevertheless a popular focus of research in synthetic biology. Principally, I am speaking of plants and animals in these chapters, though organisms such as yeast, protozoa, and even archaebacteria have many of the characteristics of the multicellular organisms. They differ from prokaryotic biology both at the cell and molecular levels. We'll go through each of these features in some detail next, so I'll just list them here. Eukaryotes have organelles which compartmentalize specialized processes. They have cytoskeletons made of actin and microtubules that allows them to change their shape. They have a G0 state that allows them to remain physiologically active while not replicating. They have gap junctions that allow them to share nutrients between cells, and finally they have programmed cell death called apoptosis that allows them to remove cells from the multicellular system. There are also differences at the molecular level. Eukaryotes have introns and spliceosomes which process mRNAs prior to translation. They have specialized degradasomes for programmatically removing proteins. They have microRNAs for epigenetic control of gene expression. They have mediator, which is a more complex means of controlling transcription initiation. The mRNAs have specialized structures at their end to signal their identity instead of relying on internal sequences for initiation of translation. And finally, they have tyrosine and MAP kinase signaling cascades for dynamic environmental sensing. One of the most salient features of eukaryotic biology is the larger number of proteins associated with familiar processes. Typically, around tenfold more polypeptides are involved in a process than are required in a prokaryote. Often these polypeptides self-assemble into large molecular machines, thus they have typically around tenfold more genes. As a result, they necessarily grow slower than prokaryotes to have time to replicate their large genomes. From a tools perspective, the genetics of higher eukaryotes is more limited than E. coli and yeast genetics. The easiest way to install genes into mammalian cells is transient transfection. Many lines of mammalian cells uptake DNA just fine, and what is challenging is getting stable replication of the genes. If you just want to express the genes for a few days, such stable replication is not required. It can be challenging to control gene dosage when using this approach. Transiently transfected DNAs will randomly integrate into the genome, and such cells can be selected with chemicals, but this is a slow, long process. There are also viral technologies that efficiently integrate into the genome. Such processes are slow to perform, but highly efficient once the virus particles are prepared. Installing more than one gene into the genome with precision is still a challenging activity with eukaryotes. Many plants are also genetically tractable. Agrobacterium is a bacterium that invades plants and delivers a large plasmid. Genetic manipulation systems derived from Agrobacterium enable users to manipulate the plant genome. Some plant cells are also amenable to gold particle bombardment strategies to introduce synthetic DNAs. Eukaryotic genetic manipulation is still far more challenging from a technical perspective than prokaryotic genetic engineering due to the slow speed of these technologies and the slow growth rates of the cells. However, the methods of gene delivery to eukaryotes is an active and moving area of research.